Hello guys, this is Jyotiswar. Welcome to my channel Study with PJs. Today in this video we are going to learn about ambivation, diffusion, osmosis and osmotic pressure. Okay, so in our previous video we have already completed all these topics, that is absorption of roots, need of water and minerals for plants, that are photosynthesis, transportation, mechanical stiffness and characteristics of roots for absorbing water. So in this video we will cover these topics. Okay, so first, what do you mean by ambivation? Okay, so here ambivation is a phenomenon by which the living or dead plant cells absorb water by the surface attraction. Okay, substances which are made up of cellulose or proteins or hydrophilic, which are also known as strong affinity for water. Okay, so examples of these are dry seeds, wooden doors swell up on contact with water or exposure to moisture air. Okay, so due to imbibitional pressure, seed coat ruptures in a case of germinating seeds. It is also an important force in ascent of sap. Okay, that is about ambivation. Now moving to diffusion. What do you mean by diffusion? So here diffusion is a free movement of molecules of a substance. Okay, the substance may be solute or a solvent or a gas or a liquid. Okay, from the region of a higher concentration to the lower concentration when the two are in direct contact. That is about diffusion. So to understand diffusion, there are some experiments in our chapter. So first we will see about experiment number 1. See here, now the experiment 1 under diffusion is, see here there are 3 beakers. Okay, so every beaker is consisting of a sugar cube or a small table of soluble dye or a crystal of potassium permanganate. Okay, all these 3 beakers are containing a small amount of any of these one. Okay, it may be a sugar cube or a small table of soluble dye or a crystal of potassium. Okay, so the sugar or the dye or the potassium permanganate slowly dissolves and spreads in the liquid eventually. Okay, so the molecules of these substances are diffused or distributed uniformly throughout the water. See here in this first beaker, the process is just now started. Okay, only the small amount is covered with it. In the second one, the diffusion is processing. Okay, in the third one, the diffusion process has been completed. Okay, to get faster diffusion complete, okay, to get a faster result of diffusion, we can take a small spoon or a iron small iron rod and stir. Okay, by this process we can get a faster diffusion. Okay, this is experiment number 1 under diffusion. Okay, so under these diffusion process, what are the definitions of diffusion applies in this case? They are, the molecules of the dye are more crowded in and near the table of the dye. These molecules move away further and further in the region. This movement contains until the molecules are uniformly distributed. Okay, as we have already discussed that stirring with a spoon or a glass rod, this makes this process faster. Okay, that is about experiment number 1. Now we will learn about osmosis. Okay, so now what do you mean by osmosis? Here osmosis is a movement of water molecules from their region of higher concentration to their region of lower concentration through a semi permeable membrane. Okay, that is osmosis. Okay, I will repeat again. Osmosis is a movement of water molecules from their region of higher concentration to lower concentration as a semi permeable membrane. Okay, semi permeable membrane. About osmosis we have already learned in our previous class, but we will revise it again. So, and another important thing to be noted from the above definition, which I said now, is that only the water molecule move from their higher concentration to their lower concentration. Okay, that is osmosis. Okay, so again, osmosis is divided into two on the basis of inward and outward concentration. So first there are two. What are those? One is endo osmosis and exo osmosis. By the name you can understand, endo osmosis means inward and exo osmosis means outward. Okay, so first we will learn about endo osmosis. Okay, first we will learn about endo osmosis. Next we will move to exo osmosis. Okay, so first, what do you understand by endo osmosis? So here endo osmosis is a inward diffusion of water. Okay, inward diffusion of water through a semi permeable membrane when the surrounding solution is less concentrated. Okay, and this tends to swell up the cells. That is endo osmosis. Now moving to exo osmosis. What do you mean by exo osmosis? Exo osmosis, we have already learned that by the word exo means outward. So exo osmosis is the outward diffusion of water through a semi permeable membrane when the surrounding solution is more concentrated. This tends to cause shrinkage of the cell. Okay, this causes a swell up of the cells, and this causes a shrinkage of the cells. Okay, so now I will explain an experiment for osmosis. Okay, to understand about osmosis in detail, I will explain you an experiment. See here, this is the experiment. Okay, what is that experiment? To demonstrate osmosis, showing testal funnel containing sugar solution. Okay, so now what do you understand by it? Okay, so take some concentrated sugar solution. Okay, take some concentrated sugar solution in a testal funnel. Okay, cover the mouth of the testal funnel with a cellophane paper, or it may be or with an egg membrane or a animal bladder. Okay, we can cover it. Now invert the testal funnel in the beaker. Okay, how as shown in the figure, invert it. Okay, invert the invert the testal funnel in a beaker containing water. Mark the level. Okay, don't forget to mark the level of sugar solution on the stem of the testal funnel. Okay, this is experimental setup. Okay, this is the setup first which you need to make. Okay, now we will understand about the control. Okay, what is the control? First I will explain you. See, this is a clamp stand. Okay, and this is the initial level. Okay, initial level we need to mark this, and this is the final level. Okay, now this the initial level to final level is called raise of level. Okay, level raise. And this is a beaker filled with water, as we have already read that, or I have explained you that this is of a sugar solution, and this is a cellophane paper, and this is the testal funnel. Okay, now what is control? So take another testal funnel with a plain water filled in it and suspend it in another beaker also containing water. Again mark the level on its stem. That is the control. Okay, by these what do we get? Okay, after a few hours the level of sugar solution in the testal funnel in the experimental setup will rise, and also the level of water in the testal funnel in the control will remain unchanged. Okay, that does not change. Okay, so three main conclusions from the experiment are: in the experimental setup, some water of the beaker has passed through the cellophane paper to enter the testal funnel containing sugar solution. Now sugar from the testal funnel has not passed into the beaker. And the third point is the cellophane paper has acted as selectively or differentially permeable membrane, okay, and it has allowed water molecules to pass through but not the sugar molecules, okay. That is about experiment to demonstrate osmosis showing testal funnel containing sugar solution. Now we will understand about, okay, now we will understand about another experiment. Before that we will understand about the modification version of experiment to demonstrate osmosis showing testal funnel containing sugar solution, okay. So but what are the slightly modifications in it are: take a more concentrated sugar solution in testal funnel and very less concentrated dilute sugar solution in a beaker, okay. The result would be same, that is the level of solution in the testal funnel would not rise. This again is because some water from the less concentrated sugar solution in the beaker would move into more concentrated sugar solution in the testal funnel, okay. That are the modification version of experiment to demonstrate osmosis. Now now see, this is an another experiment, that is rubber sheet and muslin cloth instead of cellophane as a barrier, okay. In our previous experiment we have took cellophane as a barrier, but in this we will take rubber sheet and muslin cloth instead of it, okay. That's it. There is not much changes in it, okay. Even though by the same experiment we get same answer, okay. So what are used in it are same as clamp stand, and this is the initial level and this is the final level, okay. This is the level raise, okay. This is the level raise. Now this is the capillary tube and this is binding of it, and this is a beaker, water, sugar solution in it, inside it, and this is a whisking bag. This is the knock, okay, to stop the flowing of a sugar solution, okay. Now this is about osmosis. Now we will understand about osmotic pressure. So now what is osmotic pressure? So osmotic pressure is the minimum pressure that must be exerted to prevent the passage of a pure solvent into the solution when the two are separated by a semi-permeable membrane. I think this is a big definition, but it can be simply said as osmotic pressure of a solution is a measure of its tendency to take in water by osmosis. That's it, okay. And also there are some points under osmotic pressure which we will discuss in our next video. Till then, bye bye, but don't forget to like, share and subscribe.